Okay, some joining. Hi, Harsha. Let me know am I audible. Hi, Harsha. Let me know am I audible or not.
Yes, ma'am.
Okay. So I'll be starting the class right away. If other people joins, I will be admitting them in between. Okay. Fantastic. Yeah. Could you please let me know if my slide is visible or not?
Yes, ma'am. Visible.
Okay. So today is the lecture 4. So I'll be discussing one of the previous year assignment, fourth assignment. Okay. So let's see what is the question. So the question, as you can see in the bold letter, it is written about the ABC model. So to basically answer this question, you have to have a prior knowledge of what ABC model is. So let's first see what ABC model is. Then we'll come back and try to answer this question. Okay. So if you just see a flower, there are different parts of the flower. Okay, so flower is basically the reproductive unit of a plant. It consists of different holes, like say reproductive holes and the accessory holes. Okay, so the one which are outside, that is a green color, is the sepals, and the one which here is the white color, or it could be any other color for other flowers, that is known as petals, right? So from outside to inside, if we see, so there's a sepals, petals, and this yellow color, which is that filamentous structure here, are the stamens. Okay, and then inside, or the innermost hole, is the consist of carpels, or the female reproductive part. So outside, stamens are the main reproductive part, and pistils or the carpels are the female reproductive part. Just a second, let me admit. Okay, slide is visible again, Harsha.
Yes, ma'am.
Okay, thank you. If it is not visible in between or anything, please let me know immediately, so that I can try to fix that issue. Okay, so now, as I mentioned, the different words of the flower. So the thing is, now, so when a flower is developing from, so we know that every organ develops from a meristematic cells, right? So a pool of meristematic cells give rise to a specific organ. So to but here we can see from a single maybe meristematic region, there are different holes they are arising or developing. So that means there has to have different. It has must have to get different identification, right? It might have differentiated in a different way, so that it can give rise to different holes like petal sepal. So there must be something which is specifying its identity. Hmm. So those genes, there are actually basically three so ABC class of genes whose role is to give the identity to specific organs or the floral organs. Okay, so like as what makes a sepal sepal, what makes a petal petal, what are the genes involved in it. So there is a very interesting model here, and the combination of different genes which gives the identity to its organs. So here A, B, and C, three different types of genes are there, which are basically expressed in different holes, and their combination leads to the final outcome. So the class A gene is required. So it is alone can lead to the development of sepals. Okay, and so you have to also imagine that these are going from outside to inner side. So A plus B two genes together can lead to the formation of petals. Again, B plus C leads to the formation of stamens, and C alone is capable of forming of carpel. And another interesting part is that A inhibits C and C inhibits A. So what happens is, suppose there is a mutation in A gene. So what will happen in the absence of gene A, C will now be spread in other folds as well, and as a result of which, instead of C, other organs will be formed. Like, so C we know it forms carpel. So carpel C plus B stamen. So it will be carpel stamen stamen carpel instead of sepal petal stamen carpel. And similarly, if you if you just fit into the equation, if you lose gene B, then on you're left with A and C. So A, so here only A is capable, whatever A is capable of doing it alone, that only will be reflected for the organ development. So it will be sepal carpel carpel. So the flower which will be formed finally, they will have two layers of sepal and two layers of carpels. And now if you finally lose class C gene, then as C was inhibiting A, so in the absence of C, A will be spread throughout, and now whatever the combination with B and A alone will be found. So sepal, petal, petal and sepals. Okay. So now if you see an example in the real life, you will see that when you are losing class sorry so if you're losing class B gene, then as A and B together were forming petals, okay, so there is no petal formation in this. Okay, so this is a class B mutation, so there will be no petal. So there is something known as apetal. And another, if you lose the C gene, there is formation of sepals, petals, and sepals, but no inner organs will be formed. And another thing is that the role of gene C, the C gene, is in the determinacy of flower. So what happens that when determinacy in the sense that when you are forming four holes of a flower, when it reaches to the pistil or the carpel, it should know that it should stop here. It should not produce further any other part of the flower. It should terminate there, the growth. And that is mediated by the class C gene. Just a second. Hmm. So if class C gene is not there, the determinacy is lost. So that's why what happens, that flower within a flower, something like that scenario comes in. Okay. So the C class gene mutant shows the flower within a flower phenotypic alteration. Okay. So now if we come back to the question number one, can you answer this question? Just read the question and try to answer. Hello. And you're trying to answer?
It's like both B and C class gene.
Both B and C class gene, you think so? Okay, let's see. But you remember the C from which is this matching? They have said carpal stem and carpal. So to form the sepal, okay, the first you have to remember the first one, the outer part is the sepal. Okay, to form that you require A. So definitely A should not be there. But B, you cannot lose B as well, because here, so you mean ki A and B both. Okay, A is fine, but it cannot be B, because if you lose B, then you cannot get stamens. You will get only carpels, right? Because you B plus C is giving stamen. So the answer should be A class genealogy. Okay, that's clear.
Okay, ma'am.
So now let's move to the second. You have to remember those diagrams. It will be easy to remember. Okay, so which of the following statement is false about pin proteins? So uh, have you ever heard of pin proteins? Okay, so if you have not heard, so they are the, you know about the hormone auxin, right? So auxin, to one second, let me admit. Try to join by 5 max, because in between classes I have to go back and admit. Okay, is slide visible? Slide number 4 or question number 2? Okay, so what I'm talking about is that auxin is a plant hormone. So, and any hormone we know that that is produced in a small amount, and it can go from one place to another and do its function, right? If you remember, in the human system as well. So, and it has specific functions, right? So like, as if we talk about the root development or primordial development, auxin has a important role in that. So if here in this figure, the blue dots are the auxin molecules, and this red color triangles are the transporter. So transporter molecules are something that has to it's a hormone. It needs to be transported from one cell to another. So that's why you require some proteins. Okay. So transporter. So these are the pin proteins. So what is the function of pin protein? Is that they can the function of them is that they can organize in a specific direction in the plant cell. So in this cell, if they're organizing themselves in this direction, so what will happen, that all the auxins will move from left to right side as they are organized in the right side. Okay. And another important thing is that whenever there is some transport of auxin, auxin itself can positively regulate pin protein. So what will happen? More pins will be generated in that, and the movement from left to side here is favored, but from right to left is not favored, because it's an active process. So that's why what happens, that because of this pin localization and auxin positive regulation, it leads to a flow in a certain direction. So here from left to right, you can see the where the oxygen gradient is maximum, the flow of oxygen also happening on that side. Okay, and these proteins are helping in localization or flow of the oxygen from one cell to another. And why it is that needed? Because oxygen in many times is required in a high concentration in a specific. So supposing compared to region A in compared to region B, there is more oxygen molecules. Oxygen is most likely to work in that region to have a specific role in that region. So, and that is something known as oxygen maxima. Okay, wait. Okay. Now, So this is a protein. So you just understood from now that auxin is a hormone and pin is a efflux carrier or the transporter in simple term. So what could be the false about it? And why the name is pin is because that compared to the control, this is shoot apical meristem for tomato plant. So here you can see that there are different primordia are formed. Primordia are the small the initiate the phase of the organ initiation which leads results in a mature organ, like it could be leaf or it could be flower. So it's that is known as primordium, okay, the first bud-like structure of for any organ. So compared to this wild type, where different primordia are formed for leaf or flower floral organ, in the pin mutant, where there is no pin molecules as such, or it is mutated, that forms a pin-like structure, and it does not further develop into leaves or flowers. And if you see here in the Columbia plant or the Arabidopsis plant, you see that compared to this wild type in this mutants, it the one you can see here that is giving rise to flowers, and it has previously given to leaf, which is not shown here, but here it's showing that it is giving rise to flower. But here instead of the floral meristem, it is actually just terminating and forming a pin-like structure. Okay, so the two things we understood from here. Now let's see how to answer this question. So they are asked, so before answering any question, always read the question twice, that what exactly is being asked. Like they have asked you which is the false statement. So sometimes it happens that we just hurry up and try to find true answer or something, just do some kind of mismatch. So that should not happen. Read the question twice. So which is false about pin protein. So the question number one is they can be asymmetrically localized in the membrane. Yes, we just saw that they can be asymmetrical means it is more in one side. Symmetry means symmetry is something which shows that both the side is same. But here we saw that in the right side it more pins are getting localized compared to the left side. So it is asymmetrically localized, that is correct. And when the second question is they're always placed symmetrically in the membrane. If option number A is correct, then option number B cannot be correct. And we also saw that during a flow it's not the case, right? And their oxygen affects carriers. So option C is correct. And the option D, the Arabidopsis pin mutant lacks defined SAM and it is pin shaped. Just now we saw this is correct. So option C, D, and A, these are correct, and option B is not correct. So option B is the correct answer because it's a false statement. Okay, is it clear? Anyone there?
Yes, ma'am.
Okay, please keep interacting, and as the internet connection might go wrong sometimes, so try to interact or answer if you can. Okay,
Okay, ma'am.
thank you. So let's go to the third question. Third question is visible.
Yes, ma'am.
Okay. So let's see. So here is a match. Oh, column match the columns. So here you can very simply you can understand that you have to have a prior knowledge about these proteins, right? So let's see if we have. Just now we read something in the previous class. We have discussed some of them, and let's see what we can do. So the first question option is the column A and column B. So column A is pin proteins. So pin proteins, just now we did, these are oxygen efflux carriers, right? So A2. B is oxygen 1 like OX1. So OX1 is another oxygen influx carrier. Okay, that we can see later. But C is fabulosa. So fabulosa is something that if you're not aware, then it leads to the leaf polarity. We'll see what is leaf polarity. First, let's see what are Wushel. I have discussed in the previous and other classes that it helps in the SAM maintenance along with clavata. So Wushel will go for SAM maintenance. So here, fabulosa is something which I have never explained. So I'll be explaining that here. Hmm. So if you see this leaf, this is the Arabidopsis leaf. So this side and this side, can you see any difference between these two leaf pictures?
Yes, ma'am.
What is the difference? Okay, so if you see very clear, a little bit more deep, you can see that there are some trichomes. Trichomes is the kind of hair-like structure in this. Okay, but here this is smooth. Okay, so you know about the addaxial and abaxial side. So just imagine. So just imagine this is the stem of your plant. Now this is your vertical axis. Now, and you have a leaf here. So the surface of the leaf, which is towards this vertical axis, is known as adaxial surface, and the surface which is away from the vertical axis is known as the abaxial surface. Okay, or in simple term, you can say that adaxial is the upper surface of a leaf and abaxial is the lower surface of a leaf. So lower surface is the one which basically contains stomata and all for the gaseous exchanges. Okay, and adaxial contains mainly the trichomes. And of course, there could be some color differences, etc. And adaxial is also uh, is characterized by the presence of xylem, whereas abaxial is mainly consists of phloem. So here, fabulosa is something which, so there we can see that a single leaf has two polarity. One is abduction and one is abaxial. So there has to be something which will be required to maintain this two identity. I mean this identity of two side of a leaf, right? So that some genes are involved in that. So one of them is the fabulosa. So fabulosa, what happens that whenever you have dominant mutations of fabulosa. So dominant means the more of fabulosa. What happens that it gets both the surfaces adaxial. Like there will not be adaxial abaxial. Both the surface will turn into adaxial side. Okay. So here in this mutant, you can see it's a dominant mutant where the both it becomes curled and both the sides contain trichomes. Okay, so here from as you went through this right now, it shows this class C or the fabulosa is involved with the leaf polarity, and we should previously have seen salmon. So the for question number 3, option A is the correct option as we have seen. Just I wanted to clarify this part. Okay, is this clear?
Yes, ma'am.
Okay, so let's move on to the next one. So this one everyone knows, I guess. Which of the following organelle covers the plasmodesmata in plant cells? So what is plasmodesmata? Can anyone tell? Anything from the pictures? It is here. Okay. So plasmodesmata is like a channel between two cells, so that some molecules which can freely move from one cell to another. Why? Because if you think about a plant cell, it is basically formed of the cell membrane, then cell wall, then again if you think about the next cell, again cell wall comes, then membrane. So it has to pass through a lot of layers to move from one cell to another. So instead of that, there are certain channels which makes it easy for certain molecules to move from one cell to another. Okay, so that is known as plasmodesmata, and it is anatomically made up of endoplasmic reticulum. So that network, endoplasmic reticulum networks over the cells, and this is a smooth endoplasmic reticulum. Okay, just a second, someone is asking to join. Okay, let's see. I just now explained this question previously while I was talking about fabulosa. Just see if you can answer this question. I'll read the question, you try to answer it. Okay. So leaf development displays polarity. The leaf side towards the meristem is called adaxial. The meristem, or the one I said, just a vertical axis, you can remember that. And the side away from the meristem is known as abaxial. So during normal leaf development, xylem is located towards the adaxial side and phloem towards the abaxial side. Now, what would be the probable phenotype for the fabulosa gain of function mutant, given that fabulosa gene is specifically expressed towards the adaxial side? So they are asking what will happen if you have a gain of function mutant, means more of your fabulosa is there, then what will happen, and they're giving saying that your phloem is something which is present in the adaxial side. So option number one, if you see, is the radial pattern of xylem surrounds the phloem, that will happen, or there will be radial pattern of phloem surrounds the xylem. So I have shortlisted two options. Can you try to answer which one could be the option, A or B? So what they're saying, that in the adaxial side, more of xylem is there. Okay, located towards the adaxial, and phloem is towards the adaxial. So it is on the lower surface of the leaf. Now, if you, so what we saw that fabulosa is something which is helping for the adaxial maintenance. So if more fabulosa, more adaxial character will come. So more xylem should come. That's why option A should be the correct one, because it's showing that the radial pattern of xylem surrounds the phloem. But radial pattern of phloem cannot surround the xylem because phloem is an abaxial character, and fabulosa is important for adaxialization. And here they're asking for the gain of function mutant of fabulosa, means more of your fabulosa should lead to more adaxialization, and that's why the more layer of xylem. Okay, this is clear.
Yes, ma'am.
Okay, next let's move on to the next question. So which of the following statement is false about TFL1, terminal flower 1 mutant? Okay, so the question is, which of the following is false about TFL1 or the terminal flower 1 mutant? So, for this, of course, you have to know what is terminal flower 1. So, the thing is, whenever some mutant is named, it is basically seen that whatever the phenotypic alteration happens, based on that, that phenotype, that gene is named. So, suppose here they are saying that a terminal flower 1. So why this is named as terminal flower 1? It is very obvious from the name that the mutant or the loss of function of this gene shows a terminal flower. Terminal flower in the sense that what is the case here, that whenever a flower is growing, there should be, from the shoot apical meristem it keeps on growing. Okay, it should not stop somewhere. So if your meristem here present leads to the formation of a flower and it terminates here, then further formation of other further flowers becomes difficult. Okay, or it just ends here. So it should not terminate into a flower. Instead, the meristem should keep on dividing and give rise to new flowers. But what happens in this mutant is that after some time, when a specific number of flowers or a very less number of flowers form, it gives rise to a terminal flower, because of which it does not grow any. I mean, the flowers, total number of flowers is very much reduced. Okay, so this is suggesting us that it's late. So now they have asked a false that we will see later. So it has been also seen that whenever there is a mutation in this gene, that leads to an early flowering condition. Just a second. Yeah. So that leads to an early flowering condition, and as I mentioned, that it leads to a terminal flower development that leads to a very reduced number of flower buds compared to the wild type. So this is your wild type. Call zero is something if I have not mentioned here in this class. So call zero is the wild type counterpart. So call zero is an accession on the background in which this mutation was created. So in this wild type plant itself, this mutation was created, but right now these are two different plants. This is the wild type and this is the mutant. Okay. So this mutant has a characteristic of early flowering and reduced number of flower buds. Okay. So which of the following is false about it? So as we can see, it could be sometime maybe a single terminal flower also, because it's giving rise to a single terminal flower and a reduced number of flower buds and early flowering. So late flowering is something which cannot be the right answer. Whoever is joining now, please try to join by 5:05 or 5:10, else it becomes difficult to admit in between the classes. Okay, is this clear about the TFL1 mutant?
Yes, ma'am.
Yeah. So these are some of the specific questions again. So you have to know what the function of this gene is, etc. So this kind of questions can come in your final as well. Okay. Now let's see this question. Okay. So, in an experiment to decipher the role of gene X and gene Y during plant development, the mutants were analyzed for phenotype. So, the result showed that both X and Y single mutants are defective in lateral root development, whereas the XY double mutant plant showed additive phenotype. So, which of the following would be the best conclusion for the experiment? Can anyone answer this question? Anyone? Okay. Are you trying anyone? Okay, I'll give you 2 minutes. Please try and see whatever you can answer. Then I will explain it. Anyone trying? Okay. So let's see what is saying. So here they're saying that X and Y gene, they're trying to so there are two genes X and Y. They're trying to see what kind of interaction is present between this gene X and Y. So what they're doing that they're first analyzing the single mutant. Single mutants means the if first here in gene X there is a mutation, and they're seeing suppose there is a reduction in the root length, right? Lateral root development. So just imagine that the lateral root here is reducing by minus 1 centimeter. Hmm. And for Y, suppose in Y also they are saying that compared to the wild type, it is reducing by 1 centimeter. And now, sorry, this is the mutants, okay, not the wild type. And now when they are checking the double mutant or the plant in which both the X and Y gene are mutated, they are seeing that the lateral root development is now affected by minus 2 centimeters. So what they're seeing is the additive phenotype. That means X was contributing to some development, Y was contributing to some development, and when X and Y are together or mutated together, that is leading to some developmental defect. So that is they're telling the additive. So together they're doing, but individually, separately one. So when they're coming together, their individuality is still expressed, and they're leading to minus two or the additive phenotype. Okay, so that what does that means that they are not interacting with each other, they are working in their individual pathways. That's why when they are coming together they are leading to an additive phenotype. And why they are not interacting together? Because what happens when they interact says, suppose X was something, so suppose X was something that was promoting or inhibiting Y. So what happens that if you in that scenario if you mutate X, so if you're mutating X, the function of Y, so final suppose in the rule in root development something, if you're going to mutate X, then Y cannot be activated or suppressed, so your root development will be hampered. And if you mutate Y, then also your root development will get hampered. Okay, but if you mutate both, then two things can arise. So suppose X was inhibiting Y. So what will happen that if you're mutating X, then you are seeing some defect in the root development. And now if you are inhibiting Y again, and then you may see that that initial function of this X, whatever was in the root development, that is restored. That means X was actually inhibiting Y. That's why in the presence of X mutation, if you are mutating Y as well, you are able to see the rescue or the restoration. Okay, that means these two were linked, or I mean not linked, sorry, they had some interaction even if that was a separation. In this case this was the separation, that's why you were able to see some kind of the restoration and all. But what is different here is that two genes are working in a different pathway, and when they're coming together you will see something additive, means X has some contribution, Y has some contribution, but nothing interacted, there is no interaction between them. Okay, so from here the best conclusion is that both let's see what would be the answer is, both X and Y genes have a role in root development, or both X and Y genes function in different genetic pathways during lateral root development, or both X and Y genes function the same genetic pathways, or none of the above. So as I mentioned that here they are working in a different genetic pathway, that's why you're seeing the additive phenotype. Okay, so option B is the correct answer here. And if they were in the same genetic pathway, you wouldn't have seen any additive phenotype. Instead, the original phenotype which was there, either that would have got restored, or it would have shown the same phenotype in the presence of both the mutants. Okay, is it clear?
Yes, ma'am.
Okay, let me know if anyone having any doubt on this part. Just read the question and see if you can answer anything. If you have already attended those classes, you will be able to answer it. See first, then I'll explain it. Okay. So let's see this question. Which of the following is a negative regulator of flowering locus T and suppressor of overexpression of constans 1 genes during flower development? So here is something will come known as FLC. Have you heard of FLC? This one, flowering locus C. So the TFL1, if the one which I was talking about, the one which was having in the mutant, it was having early flowering. Okay, that means if the mutant was having, just let me go to that slide, go back to the slide. Once. So here, if you remember, we saw that TFL1 was showing early flowering mutant. That means the TFL gene as a mutant is showing early flowering, the gene will have the opposite phenotype. So the TFL1 is actually as a promoter of flowering. Okay, and in regard of that, FT is another gene which actually sorry, promotes flowering, and FLC is something which is negatively regulated by FT. Okay, so it's bit of a complex. Just let me check. So here, if I draw, so this is your flowering locus T. And this is your sock one. So these two genes, if we think of them, that they promote flowering. And here FLC, what it does, that it inhibits. Okay, so this mark, when we end it with a blunt end, that means it inhibits. Okay, and whenever we show it with some kind of arrow, that means it promotes. So here, this is a circuit which you have to remember, no other way. So FLC inhibits, so which of the is a negative regulator. So negative regulator is also synonymous of a repressor. And so flowering locus C is actually inhibits or suppresses FT and SOC1, and in turn FT SOC1, which actually promotes flowering. So by inhibiting this, FLC can actually inhibit the flowering itself. Okay, is this clear? Is this clear, anyone?
Yes, ma'am.
Okay, if you have understood, now tell me what will happen if you're going to uh, mutate FLC. So, if you're going to mutate FLC, what will happen to the flowering? Will it be promoted? Flowering will be promoted or delayed? Okay, so as I just mentioned that flowering locus C is inhibiting this FT and SOC1 which was promoting flowering. So FT and SOC1 was promoting flowering and FLC was inhibiting this. So ultimately it was inhibiting flowering. But now if you're mutating FLC itself, then FT and SOC1 will not be repressed by FLC anymore and it will be do its function of flowering. So it will promote flowering. Okay, so the answer will be it will in the mutated FLC condition, flowering will be promoted. Okay, is it clear? Okay, fine. Let's move on to the next question. Hmm. So here is some. Sorry, some term is there. Vernalization. Do you know what is vernalization? Any idea what is vernalization? Okay, so vernalization is a treatment for cold treatment. Hello, anyone there? No one is answering.
Yes, ma'am.
Yeah. So do you know what is vernalization? Okay. So anyway, this FLC gene is actually sensitive to different external conditions like temperature. Okay, so vernalization treatment is a treatment of plant with cold temperature uh, like 4 degree, 5 degree, this kind of temperature. So what it happens that when you treat a plant with cold treatment like as vernalization as we are mentioning, what happens that it can induce flowering, and so even that is not the season, sometime that can lead to the flowering. So how it happens that during this cold treatment, this FLC gene, what we saw here, that FLC was actually negatively regulating flowering, right, by inhibiting some of the flowering inducing genes. So what it does is that this cold temperature negatively regulates FLC, or it suppresses FLC so that flowering can induce. So, if you okay, just now I have told what it is. Now, can you at least tell what is the false statement about this? So, which of the following statement is false about genetic pathways regulating flower development in plants? If you're not able to tell, please write in the chat box. Okay. So false statement they have asked for. So the first one is vernalization negatively regulates FLC and induces flowering. I have already told this is the true option. So if option A is true, then option B cannot be true. It positively regulates FLC. Option 3 is C, photoperiod and gibberellic acid are positive regulators of flowering. Yes, this is correct. Photoperiod in the sense the day, the length of your day and night. So you might have already heard of short day and long day. So it depends on that. And gibberellic acid is a hormone, and sometimes, or not sometimes, the cold treatment can induce gibberellic acid as well, which acts as a positive regulator of flowering. And FT and SOC1 genes are positive regulators of flowering. In the previous slide, I have mentioned that. So, as these three are the correct answer, option A, C, and D. So, the false statement is this one. Vernalization positively regulates FLC and induces flowering, because if vernalization is positively inducing FLC, then flowering cannot happen, because if FLC is a negative regulator of flowering. Okay, is it clear? Okay, let's see. The last question is that peptide signaling is an important regulator of development in plants. Which of the following are negative regulators of stomata development? So from here, at least two you can rule out is the Apitella 3 and TFL1 because these two are having the role in the development of flower. Okay, so let's see. So we know that stomata are the stomata are the pore-like structures in the cells, in the tissue, which leads to the gaseous exchange. Right. So uh, if you see under the microscope of a leaf abaxial surface, you will see that some stomata will be there, and along with other cells. So a stomata we know that is formed of two guard cells, and because of the contraction of this guard cell, the pore will be formed, and that pore is responsible for the gaseous exchange. Okay, and what important here is that it should have an optimum number of stomata so that excess water or so that excess, you know that whenever there's a gaseous exchange, water also can evaporate from the stomata. So it should not contain a very high number of stomata, otherwise it will lead the plant to face water loss, and because of the water loss the plant can go for the wilting and it can eventually die. So it should maintain an optimal number of stomata as well. So for that there are certain genes which make sure that there are not all the cells in the tissue is developing into stomata. Okay, so there are two kinds of genes are there. So one which promotes the formation of stomata, another is the which stops or regulates, negatively regulates the formation of stomata. Okay, so first one here saying that EPF1 and EPF2, actually these are the different ligands or peptides. What they do is that they can go and, so if you just imagine this violet color is the peptides EPF1 and EPF2, they can go and bind to the non-stomatal cells, and they will inhibit some other downstream molecules which will finally inhibit the formation of stomata. So this EPF1 and EPF2 will actually negatively regulate the stomata formation in other cells, and whereas stomagen, these are the molecules which helps to formation of stomata. So from here, which are the negative regulator of stomata development is this EF1 and EF2. That's all you have to remember here. Okay, is this clear? Mhm. Am I audible?
Yes, sir.
Yeah. So the questions whatever I explained or discussed today, is it clear, or do you have any doubt in any of the question? Okay, anyway this videos will be uploaded in YouTube as well. If you have any further doubt, you can go through these videos, and if you have any doubts you can ask in the comment section. We'll try to answer. Okay, so here I'm stopping the class here as there are no more questions. Yeah, thank you everyone.
Thank you, ma'am.
सुनते भी नहीं ना यूट्यूब में वैसे भी दे ही देंगे है ना तो क्लास लेने की जरूरत क्या है डायरेक्ट यूट्यूब में दे दो टेक्नोलॉजी ने सब बर्बाद किया है